Welcome, everyone. Thank you for joining us for today's Cardiogenetics webinar. My name is Dr. Casey Gifford, and I'm a member of the Betty Irene Moore Children's Heart Center at Lucile Packard Children's Hospital, Stanford, and an assistant professor of pediatrics and genetics at the Stanford School of Medicine. Our main goal at Lucile Packard Children's Hospital is to ensure that your children thrive. In this mission, we live and breathe in everything that we do, including in researching, diagnosing, and treating genetic cardiovascular disease. This is the second event in a three-part Facebook Live series where the goal is to cover different topics related to pediatric cardiovascular disease. Last week, we heard from Irene and Hannah, two of our fantastic genetic counselors, about the basics of genetics and how that could play a role in a child's pediatric heart disease. Today, we'll focus on some of the basic science research being done in the genetics space at Lucile Packard Children's Hospital and Stanford School of Medicine. And we hope that this will ultimately, all of this research will help us improve our ability to diagnose and someday treat disease. So just a little housekeeping, we'll have a Q&A at the end. So if you have any questions for me, please leave them in the comment box. And so with that, we're going to get started and talk about heart disease research at Stanford School of Medicine. We're going to start by refreshing our memory of some of the topics covered last week and how they're related to a recent Stanford team's induction in the Guinness Book of World Records. We'll then move on and discuss two of the major questions researchers are focused on in the field of pediatric heart disease and highlight some of the very specific questions researchers are focused on related to those questions. And we'll end by talking about a biobanking collaboration between clinicians and researchers and how you can get involved. And I wanted to just take a moment to emphasize that today we're discussing research. All questions you may have regarding your clinical care should be directed to our physicians. The human body contains hundreds of cell types that perform different functions necessary for life. Each organ actually contains different cells that help it to perform its function. And there are trillions of cells in your body. And your heart, for example, actually contains many cell types, including some listed here like cardiomyocytes, endothelial cells, and smooth muscle. And while cardiomyocytes might be the most famous because they provide the force necessary for the heart to beat, there are additional cell types, such as those I mentioned, that are truly necessary for the heart to perform its function. And these cells are actually capable of performing different functions because of the different genes, which are the workers in all of your cells. And as Irene and Hannah talked about, genes are encoded by your DNA, which is essentially a string of letters. And actually, if you lined up all the DNA in all of your cells, it would be long enough to go to the moon and back 1,500 times. So we have a lot of DNA in our body. And you might be wondering, how do we know what this string of letters is? How do we actually look at DNA? And so I wanted to talk a little bit about how we do that. How do we actually look at DNA? And we usually do this through a process called sequencing. We start with a group of cells and we remove or extract all of the DNA from those cells. We mix it with a couple different solutions, and those solutions allow us to make it visible or read by a sequencer, which is essentially just a really fancy microscope that can visualize the DNA. And then we use really powerful computers that can translate this information from the sequencer or microscope into a string of letters. And once this process is complete and we've sequenced all the DNA from all of your cells, this is called your genome. So now you might be asking yourself, great, but how does that help me if I'm diagnosed with a disease? And so I wanted to share one recent example of how that did help an infant treated at Lucile Packard Children's Hospital. This infant came to the hospital at 3 months of age after sudden onset of various symptoms, including seizures. There were many possible explanations for the collection of symptoms this individual had. So the physicians decided to use rapid genome sequencing to see if it helped pinpoint the cause of these symptoms. Within minutes, the patient was consented for this study, their cells were collected, their DNA was extracted, and not long after, sequencing began. And after a couple short hours, their entire genome was sequenced and a single mutation was identified that would explain some of these symptoms. And most importantly, the identification of this mutation allowed physicians to identify targeted treatments that helped these symptoms of this patient resolve. So in this scenario, they went from disease to diagnosis in hours, just a little over 8 hours. Traditionally, this takes weeks, if not months, to receive your genetic information back. So this is an amazing example of how the future of medicine really involves, in some situations, the use of genome sequencing to inform clinical care. So I just wanted to take a second to highlight this incredible team. It was led by Dr. Ewan Ashley, as well as John Gorzinski, and some of the other members are listed here on the right. And again, they were inducted into the Guinness World Book of Records for the fastest genome sequencing time. So while that was an amazing feat, sequencing can currently only help in a small number of clinical settings where we really understand the genetics of the disease. So they were able to help that infant because they knew a lot about the specific symptoms and ultimately disease that this individual suffered from. But unfortunately, when it comes to most pediatric heart defects, they fall into this category where a genome sequencing won't really inform your care immediately. So hopefully, after I say that, you're wondering, what would it take? Why can't we currently use genome sequencing to inform the care of patients with pediatric heart disease? What are the obstacles that we need to overcome to use genome sequencing to improve the diagnosis and treatment of our patients? And there are many, but I wanted to highlight two that I think are really critical to understanding why we ask certain questions in the research lab. The first is that your genome is gigantic. So as I said, you can go to and from the moon many times with all the DNA in your body. There are 6 billion letters in your genome. Every cell has 6 billion letters. And unfortunately, we don't know all of the places to look that might explain disease. So all of the differences between any two individuals, there are millions of them. And so when we're talking about understanding why someone has a disease, it's really hard to know which one might be causing disease. And that is especially the case for congenital heart disease. We really don't know where in your DNA to look or which mutations or changes in your DNA actually matter. So that makes it really hard to diagnose and then develop treatments because we don't truly know where to look in your genome. The second question that's really important and an obstacle that we need to overcome is related to which cell types actually matter in the context of congenital heart disease and other pediatric heart defects. There are some situations, such as Williams syndrome, for example, where we think we understand the cell types that are important for that disease, the cell types that are misbehaving to cause that disease, or the congenital heart defect or heart defects that might be in someone diagnosed with that disease. But in most cases, we actually don't know which cell type in your heart is misbehaving and causing the disease. There are so many cell types, some of them mentioned here, fibroblasts, endothelial cells, macrophages, that all populate your heart, and we don't know which are misbehaving. Examples where we don't know this information are congenital heart defects like hypoplastic left heart syndrome and Tetralogy of Fallot. We can diagnose them in an individual, but we don't always know exactly why they're happening and which cell types we should be looking at. So fortunately, at Lucile Packard Children's Hospital and Stanford Children's Health, we have so many incredible clinicians and counselors that are invested in these research topics. They're really interested in not just helping care for patients, but really trying to understand what's going wrong in these diseases. And more recently, a group of basic scientists, including myself, got together and formed an initiative called the Basic Science and Engineering Initiative, which is affiliated with the Moore Children's Heart Center. And this initiative was formed through a generous gift from the Moore family. And it includes myself, as well as doctors Mark Schuyler Scott, Malene Rabinovich, and Jesse Ingratz. And our goal is to really use fundamental basic research to improve the diagnosis, treatment, and care for future patients that have any type of pediatric cardiovascular disease. So next, I wanted to talk about what basic research looks like. When I say that we're doing fundamental basic research, what does that actually mean? And so I'm going to take a minute to show you a little bit about our lab, how a typical experiment might look like, or what it might look like. And so in this example, some experiments that we like to do involve cardiomyocytes. We know that cardiomyocytes are essential for your heart to function. And what we're actually able to do is culture cardiomyocytes in the lab. And when I say culture, what that means is we can grow them in a petri dish, which is here on the screen. And we actually grow them on the bottom of this plastic petri dish. And then on top of that, we put liquid. And this liquid includes the food that these cells or cardiomyocytes need to grow. And in this situation, this would be called a monolayer. So we have a monolayer of cardiomyocytes. And what we're actually able to do with these cardiomyocytes is grow them and in an incubator. And I'm going to walk you through what this looks like in just a second. And this allows us to ask lots of questions about how cardiomyocytes behave, why they might be dysfunctional, and many relevant questions when it comes to disease. So I'm going to play this video and I'll walk you through what Megha, who's a member of my lab, is doing in this video. And you're going to see an incubator, which is up here, which is where we actually grow our cells. This keeps them nice and warm and cozy and happy. We're also going to see a microscope, which allows us to look at the cells and make sure they're viable and they're happy. And then on the right, you're going to eventually see a tissue culture hood, which is where we culture our cells to keep them safe while we're changing their food or media. So here Megha is taking the cells out of this warm, cozy environment, and she's taking them out because we have to give them new food every single day. So we take them out of their warm, cozy environment. We then go and look at them under the microscope, and we make sure that they're viable, they're happy, they're healthy, and really ready to be experimented on, because we don't want to be doing experiments on cells that are not happy and healthy. We want to make sure we're truly representing proper cardiomyocyte function. And so Meg is looking in the microscope. She's making sure everything looks okay. And when she does that, she's going to move over to the right to the tissue culture hood. And this is where Megha is going to change their food in a sterile environment to make sure that these cells don't get contaminated so that we can move forward with our experiment. And so I just wanted to give you an up close and personal look at what those cardiomyocytes actually look like in the microscope. And so something that's really interesting about cardiomyocytes is they beat spontaneously. And this is related to what they do in your heart, right? Because part of the goal or function of a cardiomyocyte is to contract or beat, and it helps your heart beat. And so we can actually recapitulate that function in the lab, in our petri dishes. And so hopefully you can appreciate that these cells are contracting and moving in our dish. And what this allows us to do is ask various questions. For example, how do drugs affect a cardiomyocyte's ability to function properly? Or how might mutations that we find in a patient's genome impact a cardiomyocyte's ability to function properly? So these are some of the types of questions we can ask with these cells. What are some of the very specific questions that we're seeking to answer that are related to congenital heart disease and various types of pediatric cardiovascular diseases? So the first is related to cardiomyocytes, and it's specifically related to a defect called hypoplastic left heart syndrome. So this is a devastating congenital heart defect that's defined by an underdeveloped left ventricle, which is here on the side of this heart. And what happens in someone diagnosed with this defect is that oxygenated blood isn't actually pumped to their entire body properly. And so this is a very painful and devastating disease. And we don't know much about it. We don't know much about why it's caused, but we do know that cardiomyocytes tend to populate or live in the left ventricle and provide the left ventricle's function. And so we have a lot of questions about what might be going wrong in the cardiomyocytes in this context. For example, if the left ventricle is underdeveloped, do they multiply fewer times during development? Because individuals diagnosed with a hypoplastic left heart or HLHS, usually this presents soon after birth, which suggests to us that during development, something went wrong. And one question is, did they just not divide or grow enough times? And is that why their left ventricle is underdeveloped? Another question we're interested in is, are they less mature? Meaning, are they functioning like a cell type you might find earlier in development instead of a cell type that you really need to function once you're born? And then now you might be asking, how will that help? What's the impact on a patient if we were able to answer these questions? And a potential impact we envision, and one of the goals of our research, is can we develop a therapeutic that would encourage these cardiomyocytes to grow or mature once we know that that's the cause of this disease? And so being able to do all these experiments on these cardiomyocytes in the lab, we hope this is a question that we could answer that would eventually help patients diagnosed with this disease. Another question that we're very interested in is related to the syndrome called 22q11. And this is a syndrome that is associated with many different diseases. These individuals have a deletion on the chromosome 21, arm 11. It's a very specific piece of DNA. And these individuals often present with a collection of symptoms. And a fraction of these individuals also have congenital heart defects, including, for example, Tetralogy of Fallot. And so while we know that the deletion on chromosome 21 increases the risk for congenital heart disease because it's happening more often in this population than someone who doesn't have, or individuals that don't have this deletion, it's not sufficient. And so we're really interested in understanding what else might be going on in this person or in anyone's genome that has this deletion that might be leading to Tetralogy of Fallot. This defect in particular, which is caused or defined by a constellation of symptoms in the heart, is additionally complicated because it has so many issues. The hearts of these individuals have so many different defects. It likely involves many different cell types. This is back to this issue of which cell types are involved. And so it's additionally hard to study because we don't really know if cardiomyocytes are the right cell type to work with. And then beyond that, which pieces of DNA, if it's not the deletion on chromosome 22, which pieces of DNA are important in this disease? And so this combination of this syndrome with this defect is incredibly difficult to really dive into. It has lots of complications. But one way we're trying to do that is with a slightly different experiment where we use something called an organoid. And so in this situation, this is a big ball of cells, and it includes many cell types rather than just cardiomyocytes. And they also spontaneously beat, as in the other example I showed you. And what we hope this will allow us to do is really understand, in the situation of someone that's diagnosed with Tetralogy of Fallot, which of the cell types are really misbehaving in this disease, and which pieces of the DNA in these cells are causing the misbehavior. And we hope that while this is a very specific population of individuals that we're studying, that this research might actually allow us to identify causes of Tetralogy of Fallot in anyone that's diagnosed it, whether it's because they have a deletion on chromosome 22 or not. So we're really trying to use these experiments to improve our ability to predict or identify and diagnose disease. And the final question I wanted to introduce you to today is about the connection between the brain and the heart. So some of you may know that there is an increased risk for diseases such as autism and ADHD in children diagnosed with some heart diseases. And there's this question about whether that's genetic. Is it actually because there's a specific type of mutation or a group of mutations in your DNA that explain the high risk of the neurodevelopmental delay or something like autism in the congenital heart disease population? And so we think that it might actually be genetic. It's caused by perhaps similar genes, because similar genes have been implicated in both diseases. And this has a really important potential impact, which is that we believe if we were able to identify the mutations or differences in the DNA that are affiliated with the overlap in these diseases, that we might actually be able to predict at birth who might develop ADHD, for example. And this is incredibly important because this would suggest and allow us to identify those that would really benefit from early intervention. And I think this is a really important goal for my lab, as well as many others at Lucile Packard and the Betty Irene Moore Children's Hospital. So let's take a minute just to talk a little bit more about what some of the research initiatives are that are in development at the Betty Irene Moore Children's Heart Center. And I wanted to start by saying that research is never done on patient samples without their prior consent. And so if you happen to have been to the Heart Center, you should not worry that we did this if we didn't talk to you about this. So we always obtain consent before we do research. So at the Children's Heart Center, we're working on what we call a precision medicine pipeline. And this is where we use your genome or your genetic sequence to dictate your care, like in the example I shared earlier with the infant that had seizures. So this pipeline involves 3 steps. The first is to build a biobank. The second is to sequence genomes of individuals that are in our biobank. And then the third is to use that genome information to design experimental models similar to the ones I've been introducing thus far. And I wanted to emphasize that we've invested in this approach, and we believe a precision medicine approach is important because for many pediatric heart defects, we think the secret to disease is in DNA. It is somewhere in your genome. We just have not found it yet. And why do we believe that? Because it occurs in families, which makes it a heritable disease that likely involves DNA. So that's why we're heavily invested in the genomic sequencing space. So we're starting a biobanking initiative, and I wanted to take a second to thank the Alfred E. Mann Family Foundation, whose generous support allows this biobanking initiative to be possible. Without their support, this initiative wouldn't be happening. And so what this involves is collection of various samples from patients, including blood, and their families. And then this blood and all of these tissues that we're hoping to collect would allow us to evaluate various metrics related to your current health status, perhaps related to your outcomes, and it would also allow us to sequence your genome. And so we're sequencing DNA or your genome from patients as well as their families. And this is important because an important point I wanted to make, we're sequencing the entire genome. So this is not like a panel that Aiste and Hannah had presented where you might just sequence a couple genes. And it's not just an exome where you only sequence the genes or the pieces of DNA that encode genes, which are the workers in your cells. We are sequencing the entire genome because we don't know where to look. And so we're going to look everywhere and really increase the chances that we find something. And then the reason that we're including parents in our approach is that it's really important for us to understand how if a child is diagnosed with a disease, how their DNA differs from their parents. And so here with this first region of DNA, I'm just showing an example where if this is the child's DNA sequence, at this position, their DNA or their string of letters is the same as their father's, but it's different from their mother. And if the dad doesn't have the disease, it's unlikely that this mutation or this position in the DNA is likely causing the disease in the child. It might be involved, but it's unlikely to be sufficient or a serious cause of disease. And then another scenario is where you have a position in the child's DNA that is very different from what you see in both of the parents. And so in that scenario, that would be an area where we would want to hone in and perhaps ask whether that's involved in disease because it's different from the parents, and we would assume that the parents don't actually have these mutations. And so this is a really important component to our biobank, including family members, so we can disentangle some of these questions and differences between everyone's genomes to really hone in on the cause of disease. And then the third aspect of our pipeline, again, is to design experiments. So how can we design experiments using all of these samples and their genomes to truly understand the cell types and the pieces of DNA that are important in the disease that people are afflicted with? And so really our goal is to combine our genetic information with all these cells to try and understand the causes of disease. So our long-term goal here is really to move from survival to thriving. So thankfully, through advances in surgical interventions and clinical care, their survival rate for congenital heart disease and pediatric heart disease patients has gone up dramatically in the recent years. And really what our experiments are aimed to do in our sequencing efforts and in this pipeline I'm talking about, the goal is really to go from surviving to truly thriving and improving outcomes in the long term for patients. And some examples of this are, for example, replacing diseased tissue once. So unfortunately, many individuals need a series of surgeries. But can we use the information we're talking about and that's gathered in our experiments so that we can engineer, for example, a better valve that only has to be replaced once for the entirety of your life rather than multiple times? And can we identify tailored therapeutics? So can we use your genome sequence to identify mutations that might predict an adverse response to drugs? How can we use that information in your genome to predict that? And then how can we predict outcomes? And again, I mentioned this example of can we devise a strategy to predict, for example, autism or ADHD at birth, so that we might be able to intervene earlier in a person's life if that's something that their family decided was important. And so in closing, I just wanted to say that at your next visit, it would be great if you could ask your clinician about the biobank. If you're interested in getting involved, ask them about it. We would love to talk to you, and your clinician, I'm sure, would love to talk to you about that. And you're always welcome to follow our research. And so you can follow the research on the BASE initiative. You can also follow the research in my lab as well as in the Biobank by going through my lab's website. And so with that, I will end and say thank you for your attention. And I would be happy to take some questions. Okay. And so I do actually see questions in the box. So the first question is, what does Q11 mean? Great question. So this is in response or in reference to 22q11, which is the syndrome I brought up that includes a high risk of congenital heart disease, including Tetralogy of Fallot. So every arm of your chromosomes, which are the big pieces of DNA that you have, has a letter and a number associated with it. And so in this syndrome, individuals tend to have the arm in the specific area of chromosome 22 called Q11 that's deleted. And this area of the genome includes a bunch of genes, known genes, but it's the issue, or it's defined by this region of a specific chromosome. Next question. Are most heart centers in the country able to do research of genetic causes of congenital heart disease? I would say this is yes and no. This is a little tough to answer, but I would say that at Stanford and at the Lucile Packard Children's Hospital and at Betty Irene Moore, there's really an amazing collaboration happening between clinicians and researchers that I think is very unique. That makes us a very unique center to visit because we're all really working collaboratively to improve care. And I think that that is going to be transformative and not something that's necessarily happening at most centers in the country. Another question is, what are some interesting research findings you have uncovered recently related to heart disease? We have many interesting research findings, actually. So I'll tell you about one, which is related to a specific family that I was studying. In my research, we often use families to understand why certain individuals have or don't have disease. And often we look at families that have multiple incidences of heart disease. I mentioned we know it runs in families, and I worked with a family who unfortunately had 3 children with pediatric heart disease. And what we were able to do in this case, because the question was, how do you have 3 children with the same disease? That's incredibly unlucky and unfortunate. But it really suggested to us that it was genetic because it happened 3 times. And these individuals were all these children were inheriting DNA from the same 2 parents. And so we were actually able to sequence the exomes in this case of all of their children and the parents and identify the specific combination of mutations that we believe was causing disease. And we were able to pinpoint the causes of this disease and their phenotype using a combination of the experiments that I showed you, as well as some additional experiments using cardiomyocytes to really understand how the mutations in these patients are important for their disease phenotype. Are stem cells used in treating genetic heart disease? So this is a good question, and the answer is no. So stem cells would not be used in the treatment of heart disease. We're currently not able to create the cells that we need from a stem cell that would actually be properly functioning in a heart. Anything that's currently transplanted, as far as I know, but I am not a clinician, is not stem cell derived. There are other scenarios where you would have transplants, but these do not involve stem cells. And then I saw a question about the organoids. So how do you study the individual cells that you have in the organoids? So this is a great question because if we wanted to understand how each of these individual cells are impacting a phenotype or involved in a phenotype, we do have to find a way to separate them, right? And take apart these organoids and really look at these cells individually. And so we have a way now in the lab to actually sequence the DNA from an individual cell rather than a collection of cells. And then we also can look at the individual cells workers, or the RNA and the protein that are derived from the DNA. And we're able to look at this on an individual cellular level, which is amazing. And that's a recent advancement that's possible in the lab. And by performing these experiments on individual cells rather than this huge ball of cells, we're able to really understand and go in and ask how individual cells and cell types are involved in a disease, so that we're not just looking at a big mass of cells. And it's not, in that situation, it wouldn't be clear how we might have a phenotype if you're looking at too many cells. Another question is, what is an exome? That's a great question too. So one of the genetic tests that's can be ordered by a clinician when an individual is diagnosed with disease is an exome. And that involves the sequencing of just genes, or the DNA that encodes the genes in your genome. And the genes are what lead to the workers or the proteins that are part of your cells that perform their function. And so an exome is just sequencing of those regions. It's a very small portion of your genome, of your overall genome. So it's just protein coding. And so it's useful because we can identify mutations that we think are very likely to cause disease, but it's such a small fraction of your genome, your overall genome. And it hasn't really helped us that much to understand pediatric heart disease. So that's one reason why in the Heart Center, we're moving towards whole genome. So we're looking at your entire genome, because we strongly believe there are additional mutations outside of the exome that might be contributing to your disease. Let's see, we have many questions. Let's see, so what are some of the more common genetic heart diseases? So this is sort of a difficult question to answer. So one of the most common congenital heart defects would be a septal defect. So that's where the cells that separate either your ventricles or perhaps the two atria in your heart don't actually fuse properly, and then blood actually mixes between the major chambers of your heart. That's actually a fairly common, or the most common, congenital heart defect. There are other types of congenital heart defects. For example, hypoplastic left heart that I mentioned is much more rare. So that is far less likely to occur. Septal defects, the first type that I talked about, accounts for a very large fraction, 50 to 60% overall of the heart defects that we see. One more question. What causes a mutation? This is an excellent question. What causes a mutation? Why are there all these differences between multiple individuals? And that's because every time a cell divides, which is part of growth and part of development, and also part of homeostasis, it has to replicate its DNA. And so that every cell maintains a copy of the genome, right? That's what it needs to do its function. And unfortunately, occasionally mistakes are made during that replication process. It's not that common, And it does happen though. And usually when it happens, it's not sufficient to cause a phenotype. So there are lots of changes in your DNA compared to, the person next to you. And most of them probably don't lead to disease or involve a phenotype. But if there's a mistake during DNA replication in a place that does matter, if it's in a gene that's really important, for example, in a cardiomyocyte and critical for a cardiomyocyte to function, that's when this mutation would actually cause problems. And then let's see, are genetic heart conditions more common in men versus women? So that's a good question. Genetic heart conditions are a big umbrella term for lots of different defects. There are some that are biased. So again, hypoplastic left heart syndrome is something I obviously think a lot about. That is more common in boys. And so we see that more often in boys than we do in girls. And that actually might be genetic. It might be that boys only have one copy of their X chromosome, for example. And so maybe there are genes or mutations encoded on the X chromosome. And because they only have one copy, they don't have another copy around that's helping out, because we actually have 2 copies of every chromosome in every cell. And so there are situations where if you have a mutation in one copy, that might not be enough to cause disease because you have a good copy, a second copy that can maintain your function. But in the case of hypoplastic left heart, it might be that there's mutations on the X chromosome involved. And because boys only have one X chromosome, they have an X and a Y rather than 2 X's like in girls. That might be why there's a higher incidence of hypoplastic left heart in boys. But for lots of heart defects, it's actually pretty similar, at least pediatric heart defects. Let me see. Oh, how long does it take to do experiments in the lab? That's a great question. So the cardiomyocytes that I showed you actually took around 30 days to grow. So you can develop cardiomyocytes and get them to grow in the lab in about 8 to 10 days, but it takes a lot longer for them to mature and be ready to do the experiments that I talked about. You can also keep your cells in culture for a year. You can keep them growing in these petri dishes for a really long time and do what we call a longitudinal study, where you ask how their behavior or function might change over time. And that's something that we're doing as well. And so that's just one example. Other cell types are more or less difficult to culture, and sometimes it takes just a couple of days, other times months. And so there's experiments, the range of time for an experiment can be on the order from days to many months. So I think that that's all the time that we have today. I want to thank everyone for tuning in and for all of your great questions. And I look forward to seeing you at our next Cardiogenetics webinar. Bye.